Scan for Sovi and straight A's only. Let's go!
Trabectedin questions look intense, but this one breaks fast if you track which cells keep dividing and which cells get stuck before mitosis. First, focus on the table. Tumor cells are the ones changing after treatment, so their missing division signals matter most because the drug is supposed to stop those cells from progressing. Next, compare healthy cells with tumor cells. If healthy cells still show mitotic indicators, the drug is not generally destroying membranes or forcing every cell into chaotic division. That points to a cell cycle block in tumor cells, specifically before mitosis, when DNA must finish copying so the G2 checkpoint can be passed. Well done. Option C. Scan now, watch the signs, avoid rush. Trust given data and check steps, because the table shows where division stops.